We will proceed with the next winner of the Beutler Award, Professor Gideon Rechavi, who will talk to us on RNA modification via M6A methylation project introduction. And I will introduce Gidi. Professor Gideon Rechavi heads Sheba Cancer Research Center at the Sheba Medical Center and holds the Gerasi Chair in Oncology, Tel Aviv University. He received his MD from Tel Aviv University in 1981, PhD from Weizmann in 1987, board certified in hematology, pediatrics, and pediatric hemato-oncology. In 1992, he was appointed head of pediatric hemato-oncology in bone marrow transplantation department at Sheba. He established the cancer genomic unit, the Wiesel Pavilion for palliative care and hospice for pediatric cancer patients, and the Sheba Cancer Research Center, all at Sheba. He has served on national advisory committees for oncology and for pediatrics. He's a member of the Israel Cancer Research Fund International Scientific Council and sits on the ethics committee and of the Israeli Pediatric Hemato-Oncology Association. Professor Rechavi has recently elected to the European Academy of Cancer Sciences. He is the recipient of numerous awards, including the Kennedy Award, the Prize for Vision in Medicine, and the El-Kalas Prize for Distinctive Scientist in Medicine. Professor Rechavi received an award from the Ernest and Bonnie Beutler Research Program of Excellence in Genomic Medicine for his work on M6A RNA methylation in obesity, diabetes, and cancer. I'm honored and privileged to invite him to the podium to share with us most of his most recent research achievements.
I'm thrilled to be part of the program of the Beutler family. I don't have to introduce the Beutler family anymore. We heard amazing introduction by Jacob Rowe about Ernst Beutler, and I can tell you that each new trainee in my department in the old days, I used to give him the Williams textbook of hematology and tell him, go over it till tomorrow and tell me who wrote most of the book. And of course they came and say, amazingly, this is more than 2,000 pages book, and half of it was written by Ernst Beutler. Then I told them, now go home and learn who did the work that is described in these chapters. And indeed, it's amazing how much Ernst Beutler made in his life and in any field of hematology. Just to mention, in addition to all the contribution to iron metabolism and enzymopathies and red cells, etc., he also, his group, invented the 2-CDA, the drug that changed the therapy of hairy cell disease. And it's amazing that one person accomplished so many achievements. And therefore, we are not surprised to hear Bruce Beutler talk to see that this is in the family. So it's really a great honor to be here. And last personal note, probably the best advices that I got for my career were from Ernst Beutler, that was a good friend of my mentor, Bracha Ramot. And he, for example, is the one that suggested me a few years ago to step down from running the department and concentrate on science. So I really owe him a lot. So we heard two fantastic talks about genetics, so it's time to mention also epigenetics. And until recently, when we discussed epigenetics for the last 30 or 40 years, we discussed either DNA modifications, in particular methylation of cytosine, and we discussed epigenetic modifications of histones, those proteins that trap the DNA and control gene expression. What I'd like to suggest in this talk, that in between the DNA and the RNA there is another molecule named RNA, and we now can start to discuss also RNA epigenetics. We all read in the textbooks that there are 4 basic letters that are the basis for the genetic code in DNA and in RNA. But in real life, it's a little bit more complicated. In DNA, in addition to the conventional A, G, C, and T, there are 4 additional modified nucleotides. One of them is 5-methylcytosine, which is methylated and has to do with the well-studied DNA epigenetics. In RNA, we have 120 modifications, and it's not surprising. There are so many types of RNAs. RNA such as ribosomal RNA, tRNA, messenger RNA, and all the recently identified small RNAs, microRNAs, long non-coding RNAs, etc. And RNA is also involved in coding, in structural activities, and also in catalytic activity. Therefore, it's not surprising that the language, the vocabulary of RNA is much broader than what we realized before. In the last 10 years or so, our group was involved in the study of adenosine to inosine editing. We were the first to develop a methodology that allowed the identification of thousands and recently millions of editing sites in the human transcriptome. And over the years, many groups were able to show that it has to do with normal physiology and also with a variety of developmental disorders, neuropsychiatric disorders, cancer, etc. However, the identification of modified adenosine to inosine is very simple because the structure of inosine is very similar to that of guanosine, and therefore our routine sequencing methodology can easily identify the modified nucleotide. So you have adenosine in the DNA, you have guanosine in the RNA, you can predict that this guanosine is actually represent inosine, and therefore by sequencing and bioinformatics, we were able to uncover the human editome. I'd like today to discuss, so this is the machinery of RNA editing. I won't mention it anymore, just to say that you change A to I, which is recognized by most cellular machineries as Gs, and this can be easily identified by sequencing. I'm going to discuss another modification, which in this case it's 6-methyladenosine, turned out to be the most common internal modification of messenger RNA. However, in this case, it's a little bit more problematic to identify it. We knew about the presence of this modified nucleotide for 40 years or so, based on biochemical studies. When you degrade RNA to single nucleotides by mass spectrometry and other methodologies, it's easy to show that there are modified nucleotides, including M6 adenosine. However, there was no way to identify and to map and to quantify the level of these modified nucleotides. Over the last few years, we learned a lot about the machinery that controls the level of M6A in messenger RNA. So we have a huge complex of proteins. The best studied part of this complex is METTL3, which is the methyl transferase. This is the catalytic subunit that add methyl to adenosine in position number 6. Over the last 3 or 4 years, we learned that there are also 2 demethylases, 2 enzymes that eliminate the methyl. One is known as FTO, and the other one is ALK-BH5. And the presence of such well-controlled system indicates that we are dealing with a dynamic and controlled system that probably has important function. The the value or the importance of this machinery was shown in several model species. For example, sporulation in yeast cannot be completed without methylation. In Drosophila also gametogenesis is affected if you knock down the methyltransferase. And also in plants, in Arabidopsis, there is major defect in differentiation when you knock down the methylation enzyme. So it seems to be important, and therefore we aimed a few years ago at developing a methodology that will allow us to detect this type of modification. In the case of RNA editing, life was easy because you change the sequence and you can identify this modification by sequencing. With this modification, it's a little bit more complicated because DNA sequencing will miss this modification. So what we did, and this is mainly the work of outstanding student, Dan Dominicini, we developed a methodology to sequence, to high-throughput sequence, methylated adenosine. So what you do is simply you break the messenger RNA or the RNA to small pieces. Then you use antibody directed against the M6A. So you enrich for the fraction that contains this modified nucleotide. Then you sequence it by next-generation sequencing, and you compare it to the input, to those sequences that were not enriched by the antibody immunoprecipitation. So what you see here is a typical gene where the input, the non-selected peaks are in red. The blue peaks represent the enriched peaks, those peaks that were enriched by the antibodies, and therefore they are predicted to contain M6A, so you can locate and quantify this modification. And we managed to identify thousands of such modifications in thousands of genes, the majority of them, about 7,000 of them, in messenger RNAs, but also hundreds of modified non-coding RNAs, and I'll mention it a little bit later. We were able to show that the methylation takes place in a specific consensus sequence. So there is this purine A, CU, sequence that is enriched in the methylated region, and indeed the methylation occurs in this particular A. The next question was, can we identify the function of this modification? And I can answer you right now, we don't know yet, but the first hint towards the understanding of the function is to look for specific locations, specific positions in the messenger RNA where this methylation is found. And we were able to identify two major peaks, or two major locations. One is exactly at the end of the coding region. Here you see a schematic representation of messenger RNA. This is the coding region. The introns are marked by black lines. This is the 5' and non-translated region. This is the 3'. You see that many peaks are located around the stop coding. The second interesting finding was that among those peaks that are found in the coding region of the messenger RNA, the majority are found in very long exons. The average human exon is 170 base pairs long. The majority, 87% of the peaks that were found in coding region, in exons, were in exons longer than 400 nucleotides, suggesting that maybe this modification has to do with processing of very long exons. Maybe the routine splicing machinery needs support, needs additional localization in very long exons. I mentioned that also non-coding RNAs are affected by this modification. This includes many important long non-coding RNAs, antisense RNAs, and for example, some of the best studies non-coding RNAs, such as cyst, that control the inactivation of the X chromosome. By the way, a very issue that Ernst Beutler liked very much from the study of G6PD. We analyzed in parallel to the human methylome also the mouse methylome, and turned out that it's very, very similar to the situation in human, exactly the same consensus, exactly the same localization of peaks, and very nice conservation of methylation in mouse and human homologs, suggesting again that we are dealing with a very precise control mechanism. By applying a variety of stress and induction by interferon, hepatocyte growth factor, heat shock, UV, etc., we were able to show that although we are dealing with a quite conserved machinery, there is a set of genes whose expression is altered when you change, when you apply stress, various types of stress, and this change in methylation is also translated to the level of expression. Just for example, here's the EGF receptor. When you treat the cells with interferon, you change the level of the specific peaks. Sometimes you see new peaks or elimination of other peaks. When the catalytic subunit, the METTL3, is knocked down in hepatocytes, the major effect is apoptosis. And when you look at the pattern of alternative splicing, remember we find a lot of methylation sites in exons, there is a major change in the pattern of alternative splicing. So the first function that we can attribute to methylation of RNA is the control of alternative splicing. And when you ask in an unbiased way, which is the gene family or the pathway, the biochemical pathway that is affected in the most impressive way by change in methylation, the p53 pathway, with its many upstream and downstream components, is altered in a significant way. More than 30 proteins in this pathway are altered by alternative splicing when you modify the level of methylation. When we deal with any type of epigenetic modification, be it DNA methylation, histone acetylation, etc., we deal with writers, with erasers, and with writers. The best example is DNA methylation, the methylation of 5-methylcytosine. We have the methyltransferases, the DNA methyltransferase enzymes that add the methylation. These are the writers. Then in recent years, we learned that there are enzymes that eliminate this methylation. These are the TET proteins, and these are the eraser. And there are also readers, those proteins that bind to the modified nucleotide, or be it a modified protein in histones, and can execute the biochemical function of this modification. In the case of methylation of DNA, the best studied is MECP2. MECP2 is the protein that is mutated in patients with Rett syndrome, the severe autism that affects mainly girls. And the effect is in the readers. There are no proteins that can read in a proper way the epigenetic modification. So we ask in the next stage, can we identify the readers of RNA methylation? For that purpose, we synthesized two types of oligonucleotides. RNA oligonucleotides with the consensus sequence for methylation in two versions, with and without the M6A. And then we use these oligonucleotides to immunoprecipitate and search for proteins that bind to the methylated and not to the unmethylated oligonucleotide. In this way, we were able to identify several proteins that seems to be specific in their binding to the methylated consensus. We described in our first paper two members of the YTH proteins. There are about 70 YTH proteins in our genome, and nothing was known about them except that they are involved in RNA binding. We were able to show that YTH2, YTH3, and recently also YTH1 binds only specifically methylated RNA, and they are involved probably in a variety of cellular activities. And our colleague Chuan He very recently published in Nature the extension of our work showing that YTH2 indeed has to do with regulation of fate of messenger RNA. They were able to show that the binding of YTH2 eliminates messenger RNA from the translatable pool, takes it to the P bodies, the processing bodies, where they can be degraded and they are not translated. So this is probably the beginning of further understanding of the cellular functions of this modification. As part of the project we are involved now with the support of the Beutler Foundation, we are aiming at study the role of this modification in human diseases. Since I think 2007, several genome-wide association studies of patients affected by obesity identified a gene named FTO as the highest candidate. This was the gene that in several independent studies, very large studies, in various age groups, FTO was linked repeatedly to obesity and to diabetes. Very recently, it was estimated that about 1 million individuals in the world have a weight of, on the average, 3 kilograms more than the rest of the population due to a specific overactivity variant of the FTO. So as part of this work, we are starting to study the role of this modification in obesity. And we started by using a high-fat diet mouse model. This is in collaboration with Chaim Cohen Group at Bar Ilan University and his student. And using this well-studied model where you see the high-fat mice have overweight, the composition or the addition of weight is due to accumulation of fat. There's increased glucose level. And Shai Ben Chaim, who is working on this project, very recently was able to show that in the liver of the high-fat diet mice, there is significant overexpression of the FTO, of this demethylase that has to do with control of methylation. So now that we know that the FTO, this protein that was linked to obesity and to other diseases, but nobody knew what's its function, now we can correlate it to methylation and to look for the cellular functions of this modification. There is only one family in the world where FTO deletion or FTO mutation was identified. And luckily, this family is from Israel, from Ramle. And Orit Reich, the geneticist from Asaf HaRofeh, is collaborating with us. And we now have the cell, the fibroblasts and embryonic stem cells and the IPS of this family. And we can start using this as a model for human knockout of the FTO. In addition to obesity, FTO variants were also linked to various types of cancer. The largest work was described recently in Nature Genetics, linking the same variant of FTO that is involved in obesity with the risk for melanoma. And this association was independent of BMI. So there is even a direct link between this variant and cancer. The same was recently shown for pancreatic cancer, and very recently also to specific type of breast cancer, suggesting that indeed FTO and regulation of methylation as a new type of epigenetic modification are involved in cancer, in obesity, in diabetes, and probably more. And as part of this project, we recently defined the methodology that will allow us to quantify, to map this methylation in a more precise way. And next year, we hope to describe the fruits of this fantastic grant. Just I want to thank the group in our lab that are studying RNA methylation. It started with Dan Dominicini and Sharon Moscovich. Dan is now in postdoc with Chuan He group. Sharon is still running it with a lot of very good collaborators in our group, doing all aspects of cell biology, genomics, bioinformatics, and my coworker, Ninetta Marillo, is, of course, a major help in this huge project. And I'm really grateful to the Ernst and Bonnie Beutler program for allowing us to pursue this work. Thank you very much.
Thank you, Professor Rafi. Questions? Karl.
Thank you very much. I remember when in the 8i editing, and one of the many lectures I heard you give on that, there was an enrichment in brain-expressed genes, and you explained it by the necessity to have multiple different variants of the brain and the complexity of the brain. Is there any similar kind of observation with RNA methylation?
Yes, actually, also RNA methylation is the level of METTL3 is highest in the brain, but in particular in the hippocampus. And there are some indication that the obesity associated with FTO polymorphism has to do with increased intake. But it's only the beginning, but it's everywhere. And the demethylase, the FTO, again, is expressed all over the body. The second demethylase that was recently identified, the ALK-BH5, is expressed mainly in the testes, and the major phenotype of knocking out this specific enzyme is sterility. But for the FTO, it's everywhere, and the METTL3 is everywhere.
Yes, of course.
I just wonder about the modification near the stop codons. If you get any structural insight from looking at ribosome structure, for example, can you say whether this contributes to dissociation of the ribosome or does something else?
For sure, this is a major question. What we are doing these days is trying to correlate the level of methylation in RNA, the level of expression, and a survival of messenger RNA with ribosome profiling and with SILAC experiments to try to look for a correlation with translation. We don't have the answer yet.
Yes.
Yes.
How stable they are in the body and if they can use as a tumor marker?
We don't know. It's very simple. Actually, we are just now starting looking or mapping in a systematic way methylation in tumors. And till now, there is no study that was published mapping the level of methylation in various tumors. All we have is the predisposition, the association of this FTO, the demethylase, with the susceptibility to various types of cancer. But this is exactly what we aim to do in this project.
Yeah,
So if you see, let's say, for an RNA that's expressed in multiple cell types, do you see differential patterns, or do they have a constant pattern?
So there is basically a quite conserved pattern in different tissues, and also, as I mentioned, in human and mouse. However, when you apply specific types of stress and growth factor stimulation, interferon stimulation, etc., then you see a change of methylation in a specific group or subgroup of proteins, and also change it in level of messenger RNA expression and also alternative splicing.
Well, thank you, Professor Rafi, again for this outstanding lecture. Well, thank you all for participation for this outstanding conference. Thanks again, Prof. Beutler. And I think welcome to join all of us outside for lunch. Thank you very much.